Hello, hello.
If you need any.
Good luck.
Lighting good as well.
See if this works. Hello. Alright. Good afternoon, guys. Good evening, I should say. Can you all hear me? At the back? Alright, very good. So, welcome. Welcome back from your holidays. My name is Taha. I'm a third-year student, so currently I'm at Epworth. I started today. And I just want to preface this talk before we start just by mentioning some advice. And I think one of the main advices I like to live by in my life is I try not to take myself too seriously. That might sound odd to some of you guys, but what I mean by that is that me standing here, I'm no different to any of you sitting in the audience right now. Okay? Even though I'm presenting this talk, you might expect maybe he has some extra knowledge on this topic. But that's not necessarily true. Each of you here has knowledge specific to each of you. Each of you knows something that I don't. You know? Physiology is just one aspect of medicine that maybe interests me more, which is why I'm here giving you this talk. But I never want you to think that you can't teach me something because we can all learn something from each other. So I just want to preface this talk by saying that. So, considering everyone's here, we don't have to wait, but today we're going to have some questions. So please just make sure you have this link prepared. My alpha waves didn't work anyway. So today we're going to be talking about physiology, and mostly we're going to be going through the story of neurotransmission. Okay? The reason being, I consider neurotransmission to be a very, very important topic. Why? Because it prefaces many of the other topics in medicine, including pharmacology, anatomy, and physiology in general. If you understand neurotransmission and most of the aspects of the autonomic nervous system, it's going to give you a really good basis on understanding the other aspects of medicine. Second of all, I think neurotransmission is a bit jumbled up in how it's taught in the first year, so hopefully I can avail some of your curiosities and answer some of your questions. Okay. So part 0. First, actually, I want to show you this on the side. So what this is just a little picture that hopefully will tell you where are we at the moment in this lecture. So I'm going to begin my talk not talking about this at all. First, we're going to talk about what happens here. Then we're going to talk about what happens here. Then we're going to talk about what happens here. So it's going to go in a logical order. I'm trying to tell you a story. And stories, as you know, are usually chronological. So part 0, the resting membrane potential. So we're trying to figure out, okay, better question is, who can tell me what the resting membrane potential of a nervous cell is? Yeah, good, -70. And that changes, that resting membrane potential changes based on what cell you're talking about. For example, a skeletal muscle cell might have a resting membrane potential of -80, whereas cardiac muscle cell might have a resting potential of -90. So it varies. But neurons, usually -70. So first point to talk about is that neurons have a selectively permeable membrane. Okay? Second point is that during resting conditions, there's only one channel that's open. And what channel is that? I hear... anyone? Potassium, good. Okay. The reason I tell you that is that the resting membrane potential is most importantly depends on potassium and not sodium. The reason being is that during resting conditions, the cell is only permeable to potassium, so potassium leak channels, and not sodium. Just to help you think about it a bit more, this nervous cell is resting in fluid, and this fluid, we can think about it as a saline solution. So the fluid that the cell is resting in is pretty much sodium chloride. And inside the cell, we have potassium. And this potassium is trying to leak out, okay? And in the resting condition, this cell is impermeable to sodium. And we'll go into this in a bit more detail. Just hang on with me. So I'm just going to show you the picture. Okay. So we have this picture, right? And as you can see here, potassium is in high concentration inside of the cell and in low concentration outside of the cell, okay? And the resting membrane potential occurs because there is this uneven distribution of potassium, okay? What does a negative resting potential mean? The potential means that the electric difference between the inside of this cell and the outside of this cell is negative, okay? To explain that further, this has more negative charge than is present on the outside of the cell, okay? Now you might be confused. You're going to tell me, Taha, but look, there's 150 millimoles of potassium on this side, so how can there really be such a difference? And I'll tell you, okay? The reason, what you would expect to happen, right, because the channel is open, what you would expect to happen is potassium leaks out, correct? Because there's a concentration gradient. Are you following me so far? There's a concentration gradient here, so the question you should be asking me, why doesn't the potassium leak out, okay? To answer that question, the inside of the cell has a high amount of anions, so negative charges. Those negative charges come in the form of proteins and chloride ions, okay? And this forms an equilibrium with potassium. So, although potassium wants to leak out of the cell, and it does, for every potassium that leaks out of the cell, another negative charge becomes unveiled, and that again brings the potassium back into the cell, okay? You following so far? Good. So when something disturbs this equilibrium of potassium, then we have the movement of the ions. So say, for example, we have a mechanoreceptor that becomes deformed. That's going to disturb the equilibrium. That's going to cause sodium channels to open, and then we have a different equilibrium altogether. Okay? So in resting conditions, what I'm trying to tell you is that the main thing that allows us to have a negative 70 resting membrane potential is potassium. Okay? Any potassium that leaks out, any sodium that leaks in, we have the NAK pump, so the sodium-potassium pump, which requires ATP. Okay? And to give you a figure, about 4 millimoles you see here leaks out of the cell. That 4 millimoles is then pumped back into the cell using the sodium-potassium pump. Any sodium inside of the cell is pumped out. Okay? The other point I want to raise with you is the fact that, yes, we have a sodium-potassium pump. That requires ATP. And for that reason, the soma of a neuron is highly concentrated with the mitochondria, the powerhouse of the cell. So a neuron has a lot of mitochondria because we also have an NAK plus pump. Okay. So just to summarize what I've told you about the resting membrane potential, because this is part 0, this is just the basics of what you want to understand. The first point is that potassium is the main thing that decides our resting membrane potential. I think I'll leave it at that. So maybe we can look at it diagrammatically. So how does the potassium even leave the cell in the first place? Because we have these channels. And these, as you know, are protein channels. And protein channels, they penetrate the lipid bilayer. Okay? And protein channels can be either one of two things. They can either be voltage gated or they can be chemical gated. So when we say chemical gating, we think of a ligand. Good example, a neurotransmitter binds to the neuromuscular junction, allowing sodium to enter the cell. So that's an example of ligand binding. A mechanoreceptor deforms, causing sodium to rush into the cell. That's an example of voltage gating. So these proteins, they go through the whole layer of the lipid bilayer, and they can react to one of these two things, either electricity or to a substance. So Just give me 1 sec. I realized I put on some alpha wave music, but it's coming from my laptop. Don't worry. Anyway, so factors affecting diffusion. Okay? So there's a few things that are affecting diffusion that we saw before. I'll show you this diagram again, that we saw before. Okay? So potassium is 150 here, and it's 4 here, which is quite a big difference. So we need to understand, why does this difference exist? First one is that one of the factors that affects diffusion is a concentration gradient. So currently, potassium has quite a large concentration gradient. Do you agree with that statement? Yes. So then how do we explain this difference? And to answer that question, as we mentioned before, there's an electrical gradient as well. The inside of the cell has a large amount of negative charges that you can't see in this picture, but those negative charges push those potassium charges back into the cell. Another example of factors that affect diffusion is pressure. Although not relevant in this scenario, this would be relevant, for example, in cardiophysiology, where we have pressure that pushes fluid out of blood vessels. Okay? Okay. So we keep asking this question, why do we have a gradient at all? We've talked about the physiological reason why we have a gradient. We've talked about the sciences behind it. But what's the philosophical reason? Why did God want us to have a gradient at all? That's a hard question to answer, so I'll just try to answer it in this way. So by having a negative gradient inside of the cell, so having a negative potential, prevents spontaneous depolarization of that cell. Okay? Now think about it. There are some conditions that exist in this world where spontaneous depolarization causes debilitating syndromes. One example being epilepsy. So spontaneous depolarization allows us to have seizures. Is that something we want? No. So that's why we have a negative resting potential. And again, two things that were important was the potassium, and again this, the sodium-potassium pump. And why that was another point to remember with that is that's the reason why the soma, so the cell body of a neuron, has large amounts of mitochondria. Powerhouse of the cell. Okay, question time. So please whip out your phones or use your laptops or not, and please answer this question. Okay, that was good. And in that time that you answer that question, I'm just going to try and fix something. So has only one person answered this poll? Oh. Okay, well, everyone got the right answer then. That's good. So actually, with this, only 40 people can respond. I apologize, because it's a free account. So foxglove poison inhibits the NAK pump. Okay? Assalamualaikum. For everyone, this is my sister. So first question, what happens when we inhibit the NAK pump? What happens? We have something that was trying to change the equilibrium of these ion gradients. So when we prevent that happening, then these ions, they just naturally try and go to an equilibrium state, which is 0 on either side. Just a simple way to answer that question. Okay. Now again, we're going to finish resting memory and potential soon. But this is just another diagram that will help you to understand this. And this resource was adapted from Radner Memorial Library, thanks to them. So inside of the cell, I lost my pointer. All right, well, inside of the cell, potassium, high in force. Diffusion is pushing it out. Then at the top, low potassium, because electrostatic force is pushing it in, like we've already said. And that's an open channel. Then we have chlorine, which is a bit less relevant in this scenario. And then the last one we have is sodium. It's a closed channel. And protein again, that's the anions I was talking about. That's pushing those potassium to the inside of the cell. Okay? So we finished the resting membrane potential, which is part 0 of neurotransmission. Okay? So now we get to part 1. And again, I pull your eyes to the side of each of these slides where you see 2 neurons. And it'll tell you at what part of the presentation are we in. So currently, we're going to talk about how do we generate an action potential. Okay? So what you've talked about so far in medicine is how is an action potential generated? How does it work? So we've talked about sodium, potassium. But what we haven't talked about is how do I go from touching my hand to an action potential being generated? I hope you guys have thought of it in that way. But there's 3 main things. One can be chemical, so a neurotransmitter such as acetylcholine. The second one can be electrical. Who can give me an example of an electrical example? Anyone? What? Okay, so what I meant really... sorry. So what's an example of an organ in the body that has a gap junction? The heart, exactly. So the heart, how do you get the electrical depolarization from the top of the heart, say the top of the atrium, to the bottom of the atrium at the same time? How does that happen? The answer's there. Gap junctions. So gap junctions allow ions to transport in between the cells. And this is another thing that causes an action potential to be generated. Because again, we're disturbing the equilibrium of the resting membrane potential. Then the third one is physical, mechanoreceptors, which is something you've heard of. You've heard of it? Yeah, mechanoreceptors, good. So mechanoreceptors have been presented to you, but it hasn't really been put into the story of neurotransmission. It hasn't been explained to you in that fashion. You haven't been told why is a mechanoreceptor relevant to neurotransmission. So we'll talk about that, okay? So just to give you an example. So this is a basic example. So an example of physical sensation is somatic sensation. So mechanoreceptors are sensitive to somatic touch. Touch, feel, vibration, pressure. This is what mechanoreceptors feel. Other examples are, for example, feeling temperature, which, as you know, can either be C fibers, so free nerve endings, what I'm trying to get at, and pain sensation, which is, again, free nerve endings. So, damn, I wish I had my pointer. Okay, so let's look at this diagram and see what's happening. At this moment, I'm trying to give you an overview of how the nervous system might work. So this person is in the shower. They're feeling a temperature on their hands. Say they're feeling heat. They could also even be feeling vibration from the droplets of water hitting their hand. Okay? That sensation is felt in their skin. Then this travels up through this sensory nerve to the dorsal root ganglion. You've heard of dorsal root ganglion? And what's important about dorsal root ganglion is that it's always sensory. So dorsal root ganglion into the spinal cord. Then it rises a few levels. And then this indicates a decussation. So what does decussation mean? It switches to the other side. So it decussates. Then it rises to the somatosensory cortex. So to the part of the brain that is to do with sensation. Then the somatosensory cortex sends a signal to motor nerves. And this motor nerves goes down your spinal cord to the same level and then causes your hand to either move or something of that fashion. So that is the basic picture of what we're trying to talk about. So one of those things that can cause an action potential to be generated are mechanoreceptors, okay? And first, we're just going to talk about some definitions. So what is the function of a mechanoreceptor? very basically, you could say, yes, it's for somatic sensation, it's for touching, it's for feeling. But it goes further than that. It's also for proprioception. And proprioception means the ability of the body to realize where it is in space. So a 3D ability for the body to know at what location at a specific time it is. And how does it do that? Because mechanoreceptors are also present in our joints. It's present in our fascia. So when our fascia changes, stretch, pressure, when our joints move, then we get a feeling of proprioception because of mechanoreceptors. So location, where are these located? I've told you already, one is joints, one is in the fascia, but more importantly, on all of our hairless surfaces, which is why it's being taught to you so well. So on our palms, on our feet, and in locations where it's not reasonable to have a mechanoreceptor, we have free nerve endings, which is pretty much everywhere else. One example of that is your cornea. Now imagine, if I put my finger on your cornea, what's going to happen? You're going to feel sudden pain. You're going to blink because it's heavily innervated by free nerve endings. So that's the location. Then what does adaptation mean? Who's heard of the word adaptation? Oh, no one? You have heard of adaptation. Who can tell me what adaptation means? Anyone? Yeah, so when the nerve, or the receptor in this case, stops reacting to one stimulus and then reacts to the next stimulus, okay? So how to explain this? Say, for example, we have vibration. Vibration is something that happens very fast. So when we say something is fast adapting, this means it can switch and realize that something else is also happening. So the mechanoreceptor that detects vibration is a fast adapting mechanoreceptor because it can very quickly switch between signals. Whereas a slow one isn't going to realize that something is moving. Okay? Fast would also be an example of something moving across your skin. It stops responding to this location, then it responds to this location, then this location, so it realizes that something is moving across your skin. Then we have depth, which is nothing special about that, it's where it's located. Okay? So we have these four things, which are Meissner's, Merkel's, Pacinian, and Ruffini's. All right. And I'm sure you've been told of a way to remember this, which, to be honest, I haven't thought of a better way. So the way that people learn this is that by saying that Pacinian and Ruffini sound like very deep Italian scholars, which is why we consider them to be deep. Then the Ruffini's can be likened to roofies, which make you slow. So Ruffini's are slow and deep. The Meissner's and Merkel's, Merkel sounds like a Durkle, which is why he's slow. But a better way to remember this is that Meissner's and Merkel's are responsive to touch. If you think about it, if we want to respond to something that's touching our skin, that is going to be something that's going to be superficial. So that's how you can remember that aspect of it. So first we'll talk about Meissner's, okay? First, response to touch is an alpha-beta fiber, which just means that it's relatively fast, okay? Corpuscle means that it's a branching of terminal nerve fibers. It's located in non-hairy skin, and it's fast adaptation. So again, the example I gave is fast adaptation means it stops responding to this, and when my hand is here, then it responds to that. So it can very quickly change between stimuli, which is why you can feel something moving across your skin. So that's Meissner. So that's a picture of Meissner's, how it looks, and you can see from the diagram on the left how it does look like a corpuscle. Okay? Then we have Merkel's disc, which is also touch, but in a different fashion. It's also got alpha-beta fibers, which means it's fast. But it's a bit slower, okay? And why it's a bit slower is because it's feeling pressure. It's feeling how tightly is something touching my skin. Whereas Meissner's is testing how quickly is it moving across my skin, okay? And that's the picture of the disc. So then Ruffini's, pressure and stretch. So this is an ending. So you can imagine when the nerve fiber gets stretched, then that mechanical deformation is going to release sodium, which is going to generate an action potential. And then we have Pacinian, which is vibration. And again, coming back to that example of fast adaptation, vibration is something that we need to very quickly switch between stimuli because vibration is very fast. So that's what adaptation means, being able to switch between stimuli. And just to refresh your memory on what we mean when we say alpha, beta, alpha, delta, C fibers, etc., it refers to this. And this just refers to the degree of myelination. So how thick the myelination is, with A being the most and C being the least. Because, to refresh your memory, myelination is what allows a nerve to be fast. The less myelination it has, it's going to be slow, and therefore that confers some reasoning of its function. So if something has less myelin, then it's going to be a less important stimuli, such as pain. Okay? And to give you a better example, actually, today I'm wearing a watch. Okay? 10 minutes from now, as I give this lecture, I'm going to forget that there's a watch on my hand. Is that correct? Do you guys agree with that? That very quickly, I'm not going to realize that there's a watch on my hand. Okay? The reason for that is that touch fibers, as we just talked about, Merkel's discs, are alpha-beta fibers. Okay? So they adapt very quickly, and they no longer need to respond to a stimuli that's remaining in one location on my hand. But say I had a muscle ache. So say, for example, I tore my muscle or I stretched my muscle, and suddenly my muscle is aching. I'm going to realize that pain for the rest of the day. So it's going to be an aching, burning sensation for the rest of the day. The reason for that is that C fibers are unmyelinated. They have less ability to adapt. So that's why I can forget that there's a watch on my hand, but I can remember that I tore my, or let's say I sprained my ankle yesterday. So by understanding the physiological difference between these nerve fibers, we get an understanding of their function. Does that make sense? I see like 1, 2 nods. Everyone else is confused? I'm sorry. So we'll go on. So in the end, how does this actually cause an actual potential to be generated? You know, all I've told you right now is what are mechanoreceptors. I haven't told you how they generate an action potential. So the simple explanation is that a physical deformation, so by changing and moving the structure of the mechanoreceptor, we cause ion channels to open. So it's a mechanical opening of ion channels because, say, when I press my hand, I'm deforming my mechanoreceptors. That opens the channel, and that allows sodium to influx into the cell. And as we're going to find out today, when we have sodium, that's going to cause depolarization. Okay? So just to summarize what we've talked about, we're at part 1 of neurotransmission, where part 1 is how do we generate an action potential. We talked about 3 possibilities, one being chemical neurotransmitter, the second one being an electrical ion, for example in the heart, and the third one being physical sensation, a mechanoreceptor. So the physical deformation of the mechanoreceptor caused it to have an action potential. So we've now discussed how an action potential is generated. So let's talk about part 2, the action potential itself. So you've seen this diagram many times. Now I'm going to pick on someone. I apologize. I'm going to take a seat. What's your name, brother? Mustafa. How are you? Can you tell me what is happening in part 1? Number 1? So yeah, so the stimulus reaches the cell. And at that point in time, what's the membrane potential? And what's minus 70 mean? Resting membrane potential. Then we have a stimulus, okay? And that stimulus causes us to reach threshold, and it causes the cell to be depolarized. So one threshold is reached. And we talked about the 3 ways that happens: physical, chemical, electrical. One of those 3. So threshold is reached. Then we have depolarization. So what happens in depolarization? The sodium channels open. The sodium channels open, and they suddenly let an influx of sodium into the cell. Because remember, at the start, sodium was impermeable. Then the third part is we reach the peak of 30 millivolts. At this point in time, the potassium channels open, the voltage-gated potassium channels, and this allows the potassium to leave the cell because we're trying to get back to that resting membrane potential. So if the potassium channel opens, and remember, the inside of the cell has 140 milliequivalents of potassium. If that potassium suddenly starts to leak out, then the inside of the cell is going to become more negative, and we're going to go back to that resting membrane potential. Okay? However, we can maintain that 30 millivolts for quite some time because these potassium channels are pretty slow compared to the sodium channels. So then 4, repolarization. So at this point in time, there's something very important that happens. Okay? And I'll tell you what that important thing is. Can I ask your name? Patrick, how are you? So Patrick is going to tell me about something called refractory periods. Okay, how many types of refractory periods are there? Absolute and refractory. And that's important because at number 4, this is a point where we have an absolute refractory period. What does that mean, and how does that differ from a relative refractory period? So absolute refractory period means that the inactivation gate of the sodium is closed, I guess you could say. Okay? Inactivation gate means that no matter what you do, that sodium channel cannot be used again for this time, for this range of time, until 0.4, I wish I had my laser pointer, until 0.4, we have the absolute refractory period, which means a sodium channel cannot be opened no matter what we do. What is the reason for this? Again, philosophy, but it has an answer this time. So why do we have an absolute refractory period? To prevent an action potential from going in the opposite direction. Okay? So the resting membrane potential was to prevent depolarization spontaneously. This absolute refractory period is from preventing an action potential in going in the wrong direction. Okay? So then we have step 5, which is hyperpolarization. This means too much potassium leaves the cell. The inside of the cell becomes too negative. However, at this point, we have something called the relative refractory period. So the inactivation gate is open, but we need a greater stimulus. We need a really big stimulus to cause another action potential to be generated at this time. So that's the relative refractory period. Then step 6 is just going back to resting membrane potential, and this requires the action of the NAK pump, which again uses ATP. So, okay. Now this is the picture. You see at the bottom how we have the inactivation gate? You see this here? So this inactivation gate, during the absolute refractory period, it closes this. So this sodium channel cannot be used for whatever reason there is. During the relative refractory period, this inactivation gate is open, but this gate is closed, the activation gate. So during the relative refractory period, we just need a higher stimulus to open this gate. So do you now understand the difference between the absolute and relative refractory periods? Yeah. I see no hands, I see no heads shaking, but inshallah you did understand it. Okay. So again, back to this picture. So threshold is reached, and now look in the middle. Can you see the animation? The sodium channel open, and this allows sodium to come into the cell and depolarize it. So going back to this, the sodium channel open, and then we have this massive increase of positive charges inside of the cells, so depolarized because this sodium channel opened. And that sodium channel is going towards its equilibrium. Because remember what we said? Resting membrane potential was the equilibrium of potassium. But the sodium channel is closed, so that's not in equilibrium. So when we use foxglove poison, where these channels aren't working properly again. That's why we have it go to 0. Okay. So we've talked about part 0. We've talked about part 1, which is generation of an action potential. Now we're going to talk about how does an action potential get from the top. You see that circle? That's called the soma. That's the cell body where all the powerhouse of the cell is. So that's part 1. So we finished part 1. Part 1 was generating an action potential. And what is an action potential? So we answered those 2 questions. We answered what is an action potential? We answered how is an action potential generated? That's part 1 of neurotransmission. Then part 2 is how does this action potential go from the top of the cell to the bottom of the cell? And this is quite simple. Who can tell me actually? Who can tell me what's that straight line called from the top of the cell to the bottom? Axon. And what's an axon usually covered by? Myelin. Now what's myelin made of? What was it? Schwann cells. Well, it's not made of Schwann. It is a Schwann cell. So what's a Schwann cell made of? What's the main constituent of a Schwann cell? Yeah, lipids. So what do lipids prevent? The leaking of ions. Then you're thinking, what? How does that make sense? If ions can't move, how does the action potential propagate? Oh, we'll answer that question. So this is how an action potential works. Wow. But it's much faster than that. I mean, that was really slow. But this happens in milliseconds. And what do you call this kind of movement of the action potential? Salty conduction. No, saltatory, I'm sorry. Saltatory conduction. Okay. And how does this work? So we have to figure out how does saltatory conduction work. So this pink bit is my lipid bilayer, so my Schwann cell. And in this location, there are no sodium channels, there are no potassium channels, so no ions can leak from here. And remember, in nature, things like to go from a position of high to low. They like to go to equilibrium. So if an action potential is generated here, or it comes here, then it's going to suddenly jump to this location where there are sodium channels. Okay? So in between the myelin, there's no channel. In between the myelin, there's channels, but in the myelin itself, there's no channels. So the charge jumps like this, as we saw in that nifty animation. So philosophical questions again. Why do we need this to happen? And what's the first answer to that question? Can I ask your name? Simon. Why do we have saltatory conduction? What's the benefit? Makes it faster. So we've answered that question. So why does it make it faster? If this location was lots of channels, channels are pretty slow to open. It takes some time for them to open. It takes ions time to move from inside to outside. So instead, what we have is we just have the charges jumping across, and that's much faster. The second reason is that because we have less channels, we require less energy. And as we know, the body likes to use less energy. And this, what's this a picture of? Anyone? What's this a picture of? Can I ask your name, brother? Monty, what's this a picture of? Yeah, so it's a Schwann cell. So what I'm trying to demonstrate to you is that myelination is actually the cytoplasm of the Schwann cell covering the neuron. So this is the nucleus of the Schwann cell that keeps the Schwann cell alive. And then this is the cytoplasm, the membrane of the Schwann cell covering a neuron. So exoplasm just means the fluid inside of a nerve cell. So you can see these concentric layers surrounding the neuron. And this allows it, much like in Earth, how we have electrical wires that are insulated, this is much the same thing. So this is insulation by lipids. So we finished step 1, and we finished step 2, and now I need a drink of water. And step 3 and step 4 and there's only 4 steps, by the way, are arguably the most important steps of what I'm going to talk about today. So if you haven't been listening at this point, that's fine. You need a rest. I need a rest. So if you haven't been listening now, I urge you to please listen now, because this is the most important thing that I'm going to talk about today. Step 3 and step 4. So what we've done so far, we've talked about resting membrane potential. We've talked about how is an action potential generated and why is it generated. And we've talked about how does it get from the top to the bottom. So now we're going to talk about what happens at the synapse. So the following slides were used with permission from the Open Door worldwide website, Open Door website. So used for permission from them, so thank you to them. So this is an animation of what happens at the neurotransmitter junction. So this is a neuron, the end of the neuron in the synapse. So presynaptic neuron, this is. And those little circles are the vesicles storing the neurotransmitter. That is a calcium channel, and that will be important soon. That is the membrane of the postsynaptic cell. So postsynaptic means just after the presynaptic cell. So the top is presynaptic, bottom one is postsynaptic, simple. So purple, an action potential arrives at the buton. And buton is just the name of those little balls you see at the end of the nerves. So buton. So the action potential causes this location to become depolarized. And what does that depolarization do? It opens voltage-gated calcium channels. Calcium channels allow calcium to enter the cell. Okay? This calcium, very simply, stimulates these vesicles to bind to the presynaptic membrane. So as you saw there, is binding. Then via exocytosis, so remember these vesicles are covered by a phospholipid bilayer. When they touch the presynaptic membrane, those bilayers, they fuse together. So essentially it opens up that vesicle and allows the neurotransmitter to be released, as you can see here. This neurotransmitter will then bind to the postsynaptic membrane. So then what happens with this postsynaptic membrane, okay? Now, there are some conditions, or let's say, if we have continuous activation of a postsynaptic receptor, eventually that receptor is going to get tired, okay? So, to give you an example, how long does an epileptic seizure last? Who can answer that question? Anyone yell out a number? 5 minutes, okay. So it's a trick question, the number here doesn't matter. The point I'm trying to make is that an epileptic seizure doesn't last forever. And you have to think about why doesn't it last forever? And the answer to that question is that these postsynaptic receptors and this postsynaptic neuron, they get tired. They no longer have the energy to depolarize. Okay? But you have to remember that in that 5 minutes we were having a seizure, which itself isn't a good sign. So how do we prevent continuous activation of these receptors? We use something that's going to break down that neurotransmitter. A good example being anticholinesterase. Okay, so the neurotransmitter is destroyed by an enzyme, for example, anticholinesterase. So the anticholinesterase, that recycles the acetylcholine and it gets put back into the presynaptic neuron. So that was an example of recycling. So then this slide is decreasing quality because I made it. So this will show you, remember when I said when we have depolarization of the presynaptic neuron, calcium enters the cell. And I said very simply what calcium does is it allows the vesicle storing the neurotransmitter to go towards the towards the presynaptic membrane. Okay? And the reason I put this slide in is because it was mentioned, and I think it's important in understanding something as some pharmacology. And remember at the start of the lecture I said that the neurophysiology is very important in understanding other aspects of medicine, and this will be one example. Okay? So we have calcium. So the action potential has already come, and the calcium binds to the arms of synapse bro. That's the name of this person. So the calcium, it binds to the arms. Then what this causes this vesicle to go towards the presynaptic membrane, and it causes it to fuse. And then I ran out of time to animate, so I put that picture in. And it causes the fusion to release neurotransmitter. So let's talk about what each of these things are. So because I'm nearly certain they have been mentioned. So who can tell me what's the name of the pink arms? If you don't know, it's fine to say I don't know. Can I ask you? So synaptotagmin. You may not have heard of it, but the next two you should have heard of. Can you tell me what's the name of the red feet? Yeah. SNAP25. So therefore, what's the name of the green one? Snare. And have you heard of these things? Yeah. So the SNAP-SNARE complex. The SNAP-SNARE complex is a protein complex that allows the vesicle storing the neurotransmitter to bind to the presynaptic membrane and therefore release its neurotransmitter. And just for some questions in the future, let's think of this neurotransmitter as acetylcholine. Okay? So now that you've understood this physiological process, let's ask a question. Oh, okay, don't worry about this. Oh my lord. Okay, don't worry. So my question is, botulinum toxin, Botox. You've heard of Botox. What is why do people use Botox in general? Yeah, to get rid of wrinkles, good. So usually people will use it to get rid of wrinkles, okay? So if I tell you that Botox inhibits the SNAP25 protein and the SNARE complex, why does that prevent wrinkles from occurring? What was it? Okay, how? Can you go into further detail? Perfect. Exactly what I want. So this is a prime example of why understanding physiology, neurophysiology in this case, can help you understand some pharmacology and why people use that drug. So botulinum toxin inhibits the SNAP-SNARE complex. As we saw, the SNAP-SNARE complex allows that vesicle to bind, and that releases neurotransmitter. If we don't have release of neurotransmitter, then we don't have excitation of the neuromuscular junction. Because as you know, ACH, acetylcholine, binds to the neuromuscular junction, and that causes muscles to contract. And wrinkles is a muscle contraction, essentially. So if we inject botulinum toxin into someone's face where they have wrinkles, we prevent that contraction from occurring, therefore smoothing their skin. Does that make sense? So it's not the example I want you to remember. What I want you to realize is this is the kind of physiology that will help you understand other aspects of medicine. Okay, so this is muscle fibers, striated muscle fibers, because you see how parallel they are. And the black, like the black tree, grapes, flower, is the neuron, so the neuromuscular junction. So acetylcholine will bind to this, and it will cause this muscle to contract. Okay, sorry about this. Actually, I didn't realize this until a long time after that. I'd animated this like 4 times. So I'm sorry. So, okay. So, but just to help you, so the black things. Okay. The the black things, think of those black things are a nerve cell, right? That nerve cell has a receptor. That receptor is this receptor, okay? The red things you see there, you see those red dots? That's acetylcholine. The acetylcholine attaches to that receptor. It causes that channel to open and allows sodium to enter the cell. And then we go to part 1 of the story we've been talking about today. You have to go through it again. Okay, part 4, last part. So, to summarize what we've talked about so far today, we've talked about part 0, which is resting membrane potential. The 2 dot points to remember with that is that potassium is the main ion that decides the resting membrane potential. The second dot point is we have an NAK pump, ATPase pump. So 2 points to remember for part 0. Part 1, how do we generate an action potential? First dot point, 3 things: chemical, electrical, physical, with physical being mechanoreceptors. That's all you should remember. That's the dot point you should remember. Then, step 2 of neurotransmission was the propagation of the action potential. 2 dot points again. Nodes of Ranvier. Second is myelination / Schwann cell. Part 3, remember most of it. Sorry. Then part 4 is what we're going to talk about now, which is, okay, you see, again, drawing your eyes to this diagram. So now we're going to talk about this nerve. So we've talked about all the steps for the top nerve. So now we have to talk about the bottom nerve. Sodium. So we're going to talk about autonomic nervous system. Okay. Now this one, I'm sorry, I have to pick on people for this. So, okay. So who can tell me? Actually, first I should do this, yeah. I should ask. What are the 2 parts of the autonomic nervous system? Sympathetic and parasympathetic. And also somatic, but we don't need to talk about that today. Okay. Oh, I wish I could pick on everyone. Sorry. What's your name? Kerry. Nice to meet you, Kerry. So Kerry, for the sympathetic nervous system, and remember, if you don't know the answer, that shows bravery. Just tell me you don't know. For the sympathetic nervous system, what are the 2 main neurotransmitters? Oh, you've got it there. No, those are the receptors. I didn't realize all you guys are cheating, man. Yeah, so noradrenaline and adrenaline, okay? The reason I point that out is that, let's say 80% or the vast majority of sympathetic nerve fibers are going to respond to only these 2 neurotransmitters, okay? How about the parasympathetic nervous system? What does that respond to? Yell it out. Acetylcholine, very good. Okay, so what's the name we give to a sympathetic nervous system fiber? It's the one in blue. Adrenergic. So the majority of fibers of the sympathetic nervous system are called adrenergic fibers because they respond to noradrenaline and adrenaline. However, as we're going to find out today, the sympathetic nervous fibers can also have some other types, but we won't talk about that now. Just remember, let's put a number, let's say 80%. Let's say 80% to 90% of sympathetic nervous fibers only respond to noradrenaline and adrenaline. So acetylcholine, what's the name for those type of fibers? Cholinergic, okay? Because it responds to acetylcholine, cholinergic. Okay, what are the 2 types of receptors for the sympathetic nervous system? Alpha and beta. What are the subtypes of alpha? Well, there's lots, but alpha 1, alpha 2 are the main ones you need to know. What does alpha 1 do? I urge you to try and just remember from your memory. Because from my memories of year 1, the bad old days, all of this was very confusing for me. So this is why I've tried to put it in this kind of format so it can help you to understand what's going on. So what does an alpha 1 receptor do? Where is it located mainly? In blood vessels. In blood vessels, it causes vasoconstriction, okay? Alpha 2, this is located between two neurons. So remember in part 3 we talked about the synapse. The example we gave was the neuron and the motor end plate. That was the example we gave. But another example could be one neuron and another neuron. And the receptor on the postsynaptic neuron, instead of being the neuromuscular junction, in this case will be an alpha-2 receptor. And what does this do? It causes an inhibitory postsynaptic potential. Remember the neuromuscular junction. Those channels cause sodium to come into the cell, so therefore that's excitatory. Alpha-2, when an alpha-2 receptor on the channel is stimulated, this causes chloride to come into the postsynaptic neuron, and therefore that's inhibitory because it's making the inside more negative. Okay? So alpha-1, vasoconstriction. Alpha-2, inhibition, so IPSP. So beta fibers. These ones are a bit more important. So beta 1, the way to remember this, 1 is for one heart. So beta 1 is cardio acceleration. Beta 2, two lungs. So for bronchodilation. Now, the conceptual way to understand this, instead of rote learning it, okay? Sympathetic nervous response is trying to increase the response of everything in our body. The way that you've been taught is fight or flight, okay? Sympathetic nervous system being fight. So we want to fight for our rights. We want to fight for our survival. The way we do that is we increase light entering our eyes. So iris dilation. We increase our blood pressure so we can have perfusion to our muscles and organs. So we vasoconstrict. We increase our heart rate. So we have beta 1 activation. We increase the oxygen to our lungs so we can oxygenate our blood. So we have beta 2 activation. Do you understand what I'm saying? That there's no reason to rote learn this other than to understand where is this receptor located. The function is going to come after understanding what the sympathetic nervous system does, and the answer to that question is increase everything in the body. So parasympathetic nervous system is opposite. So we have two types of receptors, nicotinic and muscarinic, except that didn't come up with muscarinic, did it? No. That came up with NN and NM. So nicotinic receptor is also located in the synapse. Okay, remember I gave two examples. Remember, say motor end plate. The other one was alpha-2. One was excitatory, one was inhibitory. So nicotinic in that synapse is excitatory, and it's always present there. Every single synapse has nicotinic receptor. Okay, every single synapse. Then we also have NM, which was the neuromuscular junction. Okay, and then inotrope. Okay, we're not going to go into that much detail. Muscarinic. So muscarinic comes from muscarine, which comes from probably mushroom, because mushrooms activate these receptors. One thing you might know is mushroom poisoning, which causes what? Parasympathetic overactivation, which has an acronym, sludge, which causes parasympathetic symptoms because activation of muscarinic receptors. Mushroom poisoning. Makes sense, see? Conceptual understanding. M1, M2, M3. The important ones to remember is M2 is in your heart. It slows down your heart rate. If sympathetic nervous system increases my heart rate, therefore parasympathetic will slow it down. And the most important one is M3, which is everywhere. Smooth muscle constriction. That's why we give an anti-muscarinic for somebody with COPD. Have you guys heard of ipratropium? Okay, ipratropium, again pharmacology. Ipratropium is a muscarinic antagonist. Nothing to do with ants. Antagonist means it decreases the function. Because M3, constriction. If we prevent constriction, therefore we have dilation. We don't give a beta blocker to someone with asthma. Why? Exactly, because if we gave a beta blocker, we would inhibit beta 1 receptors. So we would cause someone with already small bronchioles to have even smaller bronchioles. So that's a risk. Is it an absolute contraindication or relative?
Right, we'll talk about again.
Okay, don't worry. I'm pretty sure it's absolute contraindication.
Beta blockers.
Yeah, for asthma.
Yeah.
Absolute. Okay. Another thing to help you is that some of you may have realized it by now because of what I've said. But remember, we're talking about the peripheral nervous system, i.e. the autonomic nervous system. I told you that the NN receptor, so the one in the synapse, therefore has to be in the central nervous system. So technically, the nicotinic receptors aren't part of the parasympathetic nervous system, technically. The reason I include them in this diagram is so you can understand what these respond to, which receptors is part of which system, okay? It's just important you understand that detail. I show you here. So green preganglionic, red postganglionic, nicotinic receptors everywhere. All synapses have nicotinic receptors. Some have alpha-2. Okay. And as with everything, there's some exceptions. So the sympathetic nervous system innervates all blood vessels. Remember that. Sympathetic nervous system innervates all blood vessels. So therefore, vasodilation is lack of sympathetic stimulation. Okay? Because remember, I told you alpha-1 is in the blood vessels. So if we activate alpha-1, that will constrict my blood vessel. So to dilate my blood vessel, it just means I don't activate alpha-1 receptors. Okay? There's one notable exception, which is why Helena Parkinson told you it, which is the penis. Okay? So in this case, the parasympathetic nervous system acts on the blood vessel instead of the sympathetic nervous system, which is why we have the mnemonic point and should. Point for parasympathetic, S for sympathetic. That's just one exception. Other exception is actually blushing. So when we get a blush response, so say I see a beautiful Arabic man, mutasar, sometimes I blush, and that's the parasympathetic nervous system, not the sympathetic. So I'm just telling you that always there's some exceptions, two main exceptions. Okay, again about exceptions. So this is a good conceptual understanding of how these systems work. So sympathetic nervous system, I told you, majority of those fibers are adrenergic, responding to noradrenaline and adrenaline. However, the sympathetic and 100% of parasympathetic is cholinergic. So 100% cholinergic, let's say 90% adrenergic. So we'll talk about the rest of those fibers. So sympathetic nervous system has some cholinergic fibers. It has some dopaminergic fibers. It has some postsynaptic fibers. It has some serotonergic fibers. So there's different type of fibers, but let's say, just to give it a value, let's say this is only 10% of all of the sympathetic fibers in the body, okay? So cholinergic, sweat glands and your vessels. Dopaminergic, in your kidney. Postsynaptic, your adrenal medulla. Who knows what the adrenal medulla does? I'm here, I can't hear you. Yes, it releases adrenaline into the bloodstream, okay? So how it does that is that the adrenal medulla is actually postsynaptic neurons that release the adrenaline into the medulla, into the bloodstream. So that's all I want you to take out of that. The sympathetic nervous system has some different types of fibers. Let's say 10%. This is just another way to look at it. So remember, in the synapse, every single time nicotinic. So therefore ACH. So sympathetic nervous system, adrenergic. So majority, cholinergic, dopaminergic, and postsynaptic. So different kinds of postsynaptic fibers. Now there's a sneaky receiver at the bottom. Okay. So guys, we finished all 4 parts. The story of neurotransmission is now complete. So we talked about 5 things today. We talked about part 0, which is resting membrane potential. The dot points for that was potassium is the main decider of resting membrane potential, and we have an NAK ATPase pump. Those are the 2 dot points. Then step 1 was how do we generate an action potential? Chemical, electrical, or physical. Those are the dot points you need to remember for that. Then step 2, propagation of an action potential. The 2 dot points, nodes of Ranvier, maybe saltatory conduction, and Schwann cells. Those are the dot points you need to remember. Part 4, so what happens postsynaptically? So the things to remember about that is try and conceptually understand the parasympathetic and sympathetic nervous system. So neurotransmission story is now complete. So now we'll just quickly go over a few key points about the autonomic nervous system, because this is something I'm nearly certain most of you will not understand at this point in time. Okay? So this is a cross-section of the spine of the spinal cord. Okay? But we won't go into detail about that. So the sympathetic nervous system. Now this is going to be difficult for some of you, but the sympathetic nervous system, where do sympathetic nervous fibers originate from? Which spinal levels? T1 to L2. Which part of the spinal cord? So in this picture, which color does the sympathetic nerve fibers come from? I heard someone say uncle. Green. How do you know that? Nice. So intermedial lateral nucleus, this is where all of my sympathetic fibers originate from. Okay? The thing to remember with this, why, remember, always ask the philosophical question, why? Maybe I can't answer it, but at least you ask the question. So, why is sympathetic fibers only T1 to L2? What's the reason? Why did God make me like that? I want sympathetic fibers my whole spinal cord. Why you didn't give it to me? The reason for that, the intermedial lateral nucleus only exists between, guess which spinal levels? T1 to L2. So the intermedial lateral nucleus, where my sympathetic fibers originate from, only exists between T1 and L2. Therefore, where do my parasympathetic fibers originate from? Craniosacral. So this is thoracolumbar, the red in the picture. Parasympathetic is the rest. Okay? Now, I really wish I had my laser pointer. Now guys, bear with me, we're going to finish soon. So just try and pay attention. So this is my spinal cord. Correct? What is this next to my spinal cord? What's another name for sympathetic chain? Paravertebral ganglia. The name makes sense. Para means next to, vertebral, therefore next to the vertebra, ganglia. What does ganglia mean? Collection of cell bodies. We'll explain why. Okay? Now the sympathetic ganglion might be something of great confusion for you. Well, probably not. Probably for me only. Okay, don't tell me about that. Okay. So the sympathetic chain runs the whole course of the spinal cord. Okay? A sympathetic fiber that has originated from my spinal cord can do one of 3 things, or let's say one of 4 things. Okay? I'm sorry I have to stand here, my laser pointer isn't working. So, one thing it can do, it can originate in and follow my mouse pointer. It originates in the spinal cord. It comes out here, and it goes up a level. So that's one thing it can do. And these are all pre-ganglionic neurons. Another thing it can do, it can synapse with the post-ganglionic neuron, and it can exit via a spinal nerve. Okay? The third thing that a pre-ganglionic sympathetic fiber can do, it can pass through the sympathetic chain and not synapse. Instead, it will synapse in a pre-vertebral ganglion. So these things. So those were three things that a sympathetic fiber can do. Now, there's two things called gray ramus and a white ramus. Who knows what those are? Anyone? Okay. So a white ramus is called white because the majority of fibers in there are myelinated. A gray ramus is called such because it contains much less myelinated fibers. So what is the function of each of these things? Okay? So this gray ramus and this white ramus, the only function is to connect the paravertebral chain, so the sympathetic chain, to my spinal cord. That's his function. So now we'll talk about the difference between the two. So this sympathetic fiber comes out of my spinal cord. It enters the spinal nerve, and then that fiber, it can't do anything until it goes into my sympathetic chain. How does it get there? Through my white ramus. You see white ramus? This is my white ramus. So the pre-ganglionic fiber always comes in my white ramus to connect it to my sympathetic chain. So what does the gray ramus do? So the gray ramus is for a post-ganglionic neuron. That's the difference. So just to reiterate the key points with the structure of the sympathetic nervous system, there's a few key points. First key point is that it's T1 to L2. Key point number one. Number two, all sympathetic fibers originate from the intermedial lateral nucleus, so just call it the IML. And the IML only exists between T1 and L2. That's the second key point. The third key point is that these sympathetic fibers want to get into the sympathetic chain. And they get into the sympathetic chain using the white ramus. And the white ramus is mostly myelinated fibers. So that's the third key point. The fourth key point is that post-ganglionic neurons, so these ones, the dotted lines, they go through gray rami. Okay? So that's key point number 4. And key point number 5, and the last one, is that pre-ganglionic. So these solid lines can do one of three things. One, they can go up to a higher level in this sympathetic chain. That's the first thing it can do. The second thing it can do is it can synapse inside of the sympathetic chain, and then usually the postganglionic fiber goes in the spinal nerve. The third thing that a pre-ganglionic sympathetic fiber can do is not synapse here, and actually just completely exit out of the sympathetic chain and synapse in one of these, which are so these have the names depending on which organ it innervates. For example, these are my cervical ganglia. This is my lumbar ganglia, etc., etc. Okay, and the question I want to ask you is, remember, T1 to L2 is where my sympathetic fibers come out. So another philosophical question: how does fibers that only start at T1, how do they go to my head? How is that possible? So the answer to that question is that, like we see here, that fiber can go up the chain. And remember, this chain goes the whole length of the spinal cord. Don't tell me about this, don't worry. Okay, I'll just tell you about this point. So for every pre-ganglionic neuron, on average, there's 30 post-ganglionic neurons. Okay, spinal cord and spinal tracts we're not going to go through today. Reason being, you'll learn about that eventually, okay? All I'll suffice to, well, you already have learned about it, but what I'll suffice to tell you is that the spinal cord is a very, very well organized piece of anatomy. For example, and everything has a proper name. So, my dorsal column, medial lemniscus system, or that tract, where do you think on this diagram is going to be? If I tell you that this is the back and this is the front. The back, because it has dorsal in its name. Okay? Then we have corticospinal tracts, which are located here. Pyramidal tracts. Okay? And some of these tracts will go to the medulla and they'll decussate. Okay? And this is all information I'm not going to teach you today. But you will learn, and it's very interesting. CNS, very, very quickly. So, in the CNS, we have a few types of glial cells. Glial cell just means a helper cell. Okay? 4 main types, with astrocytes, number 2, being like the 80% vast majority of all glial cells. So these are called macroglia, and compare that to microglia. So macro just means big, micro means small. So oligodendrocytes is the Schwann cell of the central nervous system. Okay? Oligodendrocyte is the Schwann cell of the peripheral nervous system, and there's a lot more oligodendrocytes than there are Schwann cells. Astrocytes, they form the blood-brain barrier. So just write down these key points. They form the blood-brain barrier, and they provide nutrients to the cell. That's what an astrocyte does. Ependymal cells, which you'll also learn this year, they produce CSF in the ventricular system. You'll learn about that more. Then microglia are macrophages. So that's the immune cell of the central nervous system. Because a lot of the central nervous system is an immune privileged location, which means it doesn't interact with the rest of the body's blood supply. Other examples being the eye and the testicles. So this picture was provided courtesy of Chairman Mackley. So this just shows you what the blood-brain barrier means. Why is it a barrier? You see, this is a normal blood vessel. It has a lot of pores. A lot of things can escape from it. This is a blood-brain barrier. It has very little gaps, very little pores. This is just for your reference. These are questions that we're not going to answer today because it doesn't work. We're not going to talk about that. I didn't cover skeletal muscle contraction. Bye.